So hello everyone who's joining our masterclass today. My name is Lea Esser and I'm part of the research and development team here at Cube Biotech responsible for protein purification and activity assays. And today I'm honored to host the Intelligent Infusion Masterclass from Cube Biotech, created for the researchers to give them the opportunity and the platform to share and promote their research with a worldwide audience. After the talk, there will be time for interactive question and answer section. You can therefore type in your questions in the lower right part of the interface with the question mark during the talk, and afterwards the questions will be shown on the screen. I'm happy to introduce our speaker today, Dr. Frank Bernhard from the Goethe University in Frankfurt, Germany. He will talk today about Cryo-EM structures of cell-free synthesized GPCR G protein complexes. Dr. Frank Bernhard is the leader of the cell-free expression units at the Center for Biomolecular Magnetic Resonance at the Goethe University in Frankfurt. After research stays in Germany, USA, and Australia, he joined as a lab leader the Institute of Biophysical Chemistry at the University of Frankfurt. The portfolio of the Cell-Free Expression Center includes the development of cell-free lysates, protein labeling, nanoparticle technologies, as well as the tailored design of membrane protein expression protocols. As a current focus, the structure evaluation is focused on the cell-free synthesized GPCRs and their complexes in lipid environment, and the concerted functional characterization of GPCRs in vitro and in vivo by applying a new nanotransfer technique. Dr. Frank Bernhard's cell-free expression system is a modular system, and the protocols can be adjusted to different targets, giving fast access to non-truncated GPCR structures in active conformation. His latest publication in Nature Communication also linked on top of the interface so that you can follow up his research. Especially, his research focus on GPCRs is really important, as GPCRs are targeted by up to 30 to 50% of clinical use drugs. So his research is focused on really hot topic. As it gets more and more clear that the function of membrane proteins is guided by the lipid environment, we are all excited to see his interesting talk today. And with this, I want to hand over to Dr. Frank Bernhard.
Yeah, thanks for the introduction, and good afternoon and good morning, and thanks for participating at my masterclass. So I'm talking today about GPCRs, which already mentioned is an important and large class of eukaryotic membrane receptors, sensing taste, smell, and a large heterogenic class of hormones, small molecules, and others. And this group is interesting for the pharmaceutical industry, and I'm talking only about members of that group. So GPCRs, once activated by an agonist, they couple to G protein heterotrimers, and these in turn activate cellular enzymes for messenger production, and so they amplify the signal which is originally perceived by the GPCR. We make the GPCRs in a cell-free system platform based on E. coli lysates, and we use a particular strategy which I will explain you in a minute. So I would like to address three different topics. So first, the production of GPCR G protein complexes, GPCR dimerization, and I also would like to introduce you into a new technique to transfer in vitro synthesized membrane proteins into membranes of living cells for the analysis in context of living cells. So first, a few words to our basic system, to our basic cell-free expression platform. So we use a two-compartment system composed out of a preparation compartment and a fixed volume of a precursor compartment. And with that, we obtain sufficient yields, a few milligrams per mil of preparation compartment. We use E. coli lysates and we produce them in-house. So I do not talk about any commercial system. We have special strains for the lysate production, but we make lysates in different ways. So we have lysates tuned for high yield. We have lysates tuned for high stability, which are enriched in chaperones. And also we use mixtures of these different lysate preparations. We operate self-expression in two formats. So in small analytical scale for protocol development. Here we have the reaction compartment up to 60 microliters in self-made reaction containers, which are then placed into the cavities of standard 24-well plates holding the feeding compartment. And once the protocol is developed, we scale up to a few mils of reaction compartment, which we operate in commercial slide-analyzers, and also they are operated in self-made plexiglass containers. So we produce membrane proteins in three different modes. So in the PCF mode, we produce membrane protein precipitates, which then need to be resolubilized or refolded. We could show crystal structures of resolubilized membrane proteins. And also in 2020, we could show the refolding of a PCF synthesized GPCR, neuropeptide Y2 receptor labeled for tryptophan. And here we could show protein dynamics upon binding or upon addition of different ligands. However, refolding of GPCRs is, I would say, a more exceptional. Refolding of GPCRs is usually not quite tricky. In the DCF mode, we also could obtain quite good quality of membrane proteins. So we showed NMR structures or the assembly of large multi-protein complexes. However, GPCRs turned out to be not very stable in presence of detergent, and also they hardly form complexes. So we switched to the LCF mode, where we add preformed membranes in form of liposomes or nanoparticle. Here, nanodiscs. And here it turned out that the insertion of the GPCRs into liposomes is also not very efficient. However, it's much more efficient if you use nanoparticles, such as nanodiscs, even if you have the same membrane composition. And we speculate here that the interface between the scaffold proteins and the membrane, which is quite heterogeneous, forms the entry sites for the translated GPCRs. So how we produce a GPCR? So we have a co-translational insertion into the preformed nanoparticle, nanodisc. You could also use solid particles. This insertion is a complete artificial way, potentially caused by simple hydrophobic interactions. So we do not use translocon signal sequences or any microsomes. Certainly, the insertion might, depending on the target, have a preferred membrane composition. So we screen a certain library of preformed nanoparticles assembled with different lipids and lipids combinations. And what you see here, it's basically a modular system. So the basic cell-free system containing precursors, energy system always stays constant. And what we change, depending on a new target, is simply the DNA template. And depending on the complex you want to produce, the G protein or the specific particular agonist or antagonist. And if available, also stabilizing nanobodies or antibodies. Benefit is that you always have complete control of the supplied compound, so you exactly know what is the concentration of your ligand, of your agonist, of the G protein. Anything you add, you don't have to worry about transport or degradation of your compounds. What you add is right away at the ribosome and can interact with your freshly synthesized protein. There are quite many GPCR structures published and available. but many of them are heavily engineered. So they have truncation of the EL3, of the terminal domains. They are expressed as fusion with large fusion partners, or they have insertions of stabilizing proteins like T4L. And this is a very important difference to our self-resistance. We always synthesize the full-length protein. And it's only modified by a short C-terminal purification tag, a strep tag or a his tag, and by a short N-terminal initiation of translation tag, which is up to 6 additional codons. In addition, the preparation, for example, of cryo samples is fast. Yeah, we incubate overnight and make purification next morning. So if we would set up now a reaction tomorrow after lunch, we could start to prepare the Cryo-EM kits. So I'll talk about 3 different GPCRs, histamine receptors, adrenergic beta-1 receptor, and free fatty acid receptors. All are from human, all are full-length receptors. Certainly they all have key roles in prominent human disorders or diseases. And also what you should keep in mind, I talk about the histamine 1 and 2 receptors and the free fatty acid 2 and 3 receptors. And these are forming dimers and also homodimers and also heterodimers. And I show you later that this formation is also possible in the cell-free system. So this is a general preparation strategy for Cryo-EM samples. So we have 2 milestones to achieve. First, the yield. This is we optimize in the PCF mode, and our benchmark is to achieve about 1 milligram per mil of expression. If you have a good working system, like the E. coli system, you achieve this quite fast, maybe in a couple of weeks you have that. Second, we try now to solubilize the synthesized GPCRs by addition of preformed nanodiscs. And here we have our library of nanodiscs assembled with different levels that you see here. This test we usually perform with GFP or MNG fusion just for convenience. This screen is not so extensive as it might look like. Yeah, usually negatively charged lipids like PG are the most effective. I would say in 80% of the cases for GPCRs, we use either DEPG or DOPG lipids. And then you screen a little bit about DOPC, or if you have the addition of cholesterol or other lipids might change anything. Then you have the solubilization of your GPCR, but the point is what is solubilized must not be active. And for the quality, we highly recommend to perform an additional size exclusion profiling. And what you see here is that you have significant fractions at or close to the void volume. And this is solubilized GPCR. In that case, it's a histamine 2 receptor. And we think here that these are particles which are simply not completely inserted into the nanodisc, so just maybe by 1 or 2 ms. The rest is sticking out of the nanodisc, and these particles have absolutely no chance to fold in a functional form. So the interesting and folded fraction you see here, this is completely integrated, and you can see that by addition of diverse ligands, you can even modulate the amount of these folded fractions. That is a histamine 2 receptor. Here you see the profile without any ligand. Here you see quite a little folded fraction. Here is with the agonist histamine. And here is with a variety of different antagonists. You see that the antagonists have a much better effect on the quality of the histamine 2 receptors than the agonists. Nevertheless, as we wanted to make cheap protein complexes, we finally selected this condition. If available, you can further check in this fraction of completely integrated GPCRs what is the amount of ligand binding activity if you have a radioassay on hand. And then you can go ahead, upscale to a few milliliter of cell-free reaction, which is usually not a problem. You add your compounds you need for the complex formation. And then overnight incubation, next day you make a purification based on the strep tag or his tag, size exclusion. Then you take the central fraction of the size exclusion, you concentrate down to around 30 microliter with a concentration you should achieve in between 2 and 3 mg per ml. And then you need about 3 microliter of that per grid. So this is, when you start with 2 mils, yeah, this is completely sufficient to prepare a whole set of Cryo-EM grids. So in case of the histamine receptor, this was as it looked like. Here is a representative micrograph of one grid. A good thing for the nanodiscs is they do not have any preferred orientation on the grid. So they have random orientation, so you can collect a lot of different class averages as shown here. And this makes structure calculation quite easy. With the histamine 2 receptor, we came down to a resolution of 3.4 angstrom, which is reasonable. There was one cryo-EM structure published before of the histamine 1 receptor complex with GQ protein with a similar resolution. There are some other structures of inactive histamine receptors, and if you see, yeah, they all contain, yeah, some deletions of terminal domains or of internal domains or some insertions. So what we have here is the first structure of a full-length histamine receptor. It was also the first structure of a self-synthesized GPCR and the first GPCR-GS complex in the context of nanodiscs. The structure has some interesting details. I just show you one. We have 4 histamine receptors. All 4 recognize histamine, in addition to a variety of receptor-specific ligands. So histamine was known. It is coordinated by TM3 and TM6 by highly conserved residues. Also, these residues coordinate the histamine in the histamine 2 receptor. It was unclear whether TM5 is involved in histamine coordination because this was observed in the structure of the histamine 1 complex. We could show that in the histamine 2 receptors, this TM5 plays not a role in the coordination of the common agonist histamine. However, this residue is a key residue in the coordination of ligands that are specific for the histamine 2 receptor. So this is a kind of a selection residue for the histamine receptor and so might become of pharmacological importance. So a second structure I would like to show you, and this is not published yet, is of the full-length human beta-1 adrenergic receptor. The beta-1 adrenergic receptor is probably the most well-analyzed GPCR, and I guess more than 20 structures have been published. However, all structures, all but one, are from Turkey and not from the human receptor. So only structure of the human beta-1 adrenergic receptor was published here in 21. And it's not in complex with the G protein. It's rather stabilized by a nanobody, and also it contains insertion of the T4L. All so far published structures of beta-1 adrenergic receptors are heavily engineered by truncations of the terminal domains, by the internal loop 3, and even also by some insertions. So this is the first structure of a full-length beta-1 adrenergic receptor. If you compare the sequence of the human and the Turkey beta-1 adrenergic receptor, you see here the yellow highlighted residues are the different residues. And exactly those residues are in the terminal domains and in the EL3, yeah, and usually they are deleted. So if you look now in our structure, where are these residues of the full-length receptor? Here we have the helix 8, so here we would expect the C-terminal domain. Here is the TM1, so here we would expect the N-terminal domain. That is the TM5, so here in between TM5 and TM6 we would expect the EL3. You don't see anything. So these additional domains are highly flexible. They are not ordered, so we could not calculate the structure. However, because they are there, they have significant constraints on the adjacent domains. You already see this in this overall structure, that the TM5 of the full-length receptor is much longer than here in the truncated receptor. And if you have a closer look of the interface of the GPCR with G alpha or G beta, which you see in the next slide, so you see quite a significant difference. Here we have the truncated version, and you see what you see here, the white lines are polar contacts up to 3 angstrom. And you instantly see that in the full-length receptor, you have many more contacts than in the truncated version. The elongated TM5 forms a new cluster of interaction with the G alpha here and here. And also we have with the elongated helix H, which has another orientation because of the constraints of the attached C-terminal domain. We have contacts even to G beta, which is very rare observed in G protein, in GPCR G protein complexes. So indeed, yeah, it makes a difference whether you analyze a full-length or a truncated receptor. And those small, yeah, so we don't have changes in the overall structure, but we have structures in the details. And those small details might have any biological significance, of course. So let's switch to another topic, GPCR dimerization. So what we try to do is to try to make structures of GPCR dimers. This dimerization is a tool of the GPCRs to expand the functional portfolio. So GPCR dimers could be homo or heterodimers. They usually have differences in ligand binding affinity and also in the signaling capability. So the function of dimers is often different from that of the monomers. We could already observe dimers here in our single particle analysis. Here is also beta-1 adrenergic receptors. So we have 2 GPCRs in one nanoparticles, obviously also coupled to two G protein complexes. Certainly, this does not say that they are dimers. It could be that just occasionally we have 2 insertions in the same nanodiscs. So we try to better analyze the dimerization here with the example of the free fatty acid 2 and 3 receptors. Also with those, we could form G protein complexes by the protocol I showed you before. So here you see complexes of the FF2 receptor with GI heterotrimer. We did not proceed with that for structural analysis because the structure came out beginning of the year, so we rather focused on the dimerization. And to show that we really have a dimerization, we implemented native mass spectrometry, where the complexes are dissociated by increasing laser beams. And you can see here nice signals of the dimer in the nanodisc or attached with a scaffold protein, with a dimer, a scaffold protein dimer, with a scaffold protein monomer. And most important, you also see the dimers without any scaffold protein, here for the FF2 receptor, here for the FF3 receptor. This proves, yeah, basically that we have real dimers and not artificial dimers somehow induced by contacts to the scaffold protein or by anything else of the nanodisc complex. We further wanted to analyze the dimerization by pull-down assay, and here we implemented a new and very, to my opinion, very helpful technique initially invented or reported by the group of Matthew Coleman. It's a nanotransfer of membrane proteins into the membranes of living cells. Yeah, there are plenty of techniques, strategies published to transfer soluble proteins into a living cell, but almost nothing, yeah, where you can transfer membrane proteins into living cells. So this nanotransfer, which is very easy, is basically a new technique that opens you a lot of possibilities. It's very simple. What you do is you incubate the living cells with a purified sample of GPCR in nanodiscs, then the two membranes align to each other. The scaffold protein is taking off, and anything which is inside of the nanodisc, so the lipids and also the GPCR, or it could be any other membrane protein, is inserted into the cell membrane. That's some basic characteristics. So this transfer is very fast. Within 1 hour, you get already a significant transfer. You can nicely tune the amount of transferred protein by incubation time or by concentration of your protein solution. The transfer is independent of the lipid composition of the nanodisc membrane. It's also independent of the type of the inserted membrane protein. Here we tested the transfer of different GPCR and other membrane proteins. So it's more a universal technique. If you compare to classical transfection, we have also quite some differences. Transfection is more efficient, so the efficiency of the transfer is about 10% to that of the transfection. However, transfection is much more heterogeneous. So in the cell population, in the transfected cell population, you have individual cells that express a protein. In that case, we have the endothelin B receptor. Like L and others don't express at all. And here with the transferred cell population, you see that more or less all transferred cells have more or less the same amount of protein. Very interesting is that with a nanotransfer, you have access to primary cells. So what you see here is a freshly isolated hard muscle cell, and it's transferred with a histamine 2 receptor attached to MHE for better visualization. It's not possible to transfect those cells because we cannot cultivate them. Within 1 day, they lose their differentiation, and transfection takes even longer. So those primary cells are only accessible by the nanotransfer for the analysis of recombinant protein. Okay, but back to the dimerization. So what we did here with a free fatty acid receptor, first we checked whether they all transfer, which here is the case, FF1, 2, and 3. So what we did, we first transfected the cells with templates, suitable templates, synthesizing the GPCRs. And then we transferred into these transfected cells combinations of our self-free synthesized and purified GPCRs. So the goal was to identify dimers between the self-free synthesized transferred GPCR and the transfected and cellular synthesized GPCRs. These contain a flag tag for identification, and the self-free synthesized GPCRs, they contain a strap tag for purification. So what we next did, we lysed the cell after the transfer, made a strap tag purification, which you can see here. And then we checked via a flag tag antibody whether we have pulled down the cellular GPCR synthesized after transfection, and indeed this is the case. So you see here transfected FFR2, and this is a pull down of transferred FF2. So this is a homodimer. Transferred FFR3. So this is a heterodimer, and here vice versa. We do not have any pull down with transferred FFR1, and this is as it should be, because the FFR1 does not recognize short chain fatty acid, but long chain fatty acid. And it's known that it has nothing to do with a free fatty acid receptor 2 or 3. That is the same done with the histamine 1 and 2 receptors. Again, here you have the transfer with MHE derivatives. If you add agonist histamine, you also see that they are internalized. So obviously the transferred self-free synthesized GPCRs are also phosphorylated. And again, here we made the pull down with previous transfection of the cells, pull down with a strap, and again we have a pull down of the transfected and cell-based synthesized GPCRs. So here you see the homodimer of HR1 self-free synthesized non-transfected. Here's a heterodimer, self-synthesized histamine 2 receptor, cell-based synthesized histamine 1 receptor, and here vice versa. And here we used as control the FF2 receptor. Just showed, yes, that it nicely dimerized with itself and with the FF3 receptor. And here, yeah, you don't have any pull down because it's clear, yes, the FF2 receptor does not interact with the histamine receptors. So this nicely shows, yes, that self-synthesized GPCRs have a very nice folding and can interact even with cell-based synthesized GPCRs. So that's already at the end of my talk. Just as a summarize, yeah, what I can, as a kind of take-home message. Anything I've shown was made in E. coli lysates, so obviously they are nicely suitable even for complex eukaryotic membrane proteins and complexes. Showed you 2 Cryo-EM structures of GPCR G protein complexes, formation of GPCR oligomers. And I introduced you into the nanotransfer, where you can simultaneously analyze membrane proteins in vitro and in vivo. Yeah, so you just make 2 oligos, one you throw on your cells, the other one you make mass spectrometry or radioassay or whatever you want. Yeah, and I'm sure, yeah, that this technique will also have a number of many new applications. And at the end, I would like to thank our collaboration partners from Frankfurt University, in a group of Nina Morga, who helped us with the native mass spectrometry. These people helped us with the Cryo-EM structure evaluation of the GPCRs. Felipe Merino helped us with the Cryo-EM structure of the beta-adrenergic receptor. Philipp Sasse provided us with primary cells, and these are our funding organizations. And I thank you for attention.
Yeah, thank you for the great talk. And now we are coming to the interactive question and answer section. Waiting for the first question is then, was the NB necessary to obtain this structure? If the NB is not there, is the structure too flexible to obtain clear information?
The structures I've shown you are with nanobody 35. Our strategy was nanobody 35 contained a histag. Our strategy was even to finally purify the complex via this histag of the nanobody 35. So with that strategy, indeed it was necessary. We did not attempt to make it without the nanobody. Theoretically, it is possible. We could purify the complex also via the strep tag of the GPCR. But the nanobody 35 is known to stabilize the G protein trimer. So, yeah, and it also makes the complex larger, so which would facilitate the Cryo-EM evaluation. So it has benefits. Yeah. Also, I don't know whether it is absolutely essential. Yeah.
Then coming to the next question. So first, he thanked you for your nice presentation. And the question is regarding the controlling of the right size oligomeric state of the protein from size-excluding chromatography fractions. It is often not easy to get the right size of the detergent-soluble proteins. Do you have any advice for the beginners?
Yeah, our strategy is to co-translationally insert the GPCRs into the nanodiscs. During that process, the nanodiscs have a fixed diameter, 10 nanometers. And during insertion, a certain amount of the lipid is kicked out. We showed this in a previous paper by NMR. So whether you have the insertion of a GPCR monomer or GPCR dimer or whatever, would not change the hydrodynamic radius of the nanodiscs. So from size exclusion, you cannot tell whether you have a monomer or an oligomer in the nanodisc. because the diameter does not change. What you can tell, and this I showed you in this size exclusion profiling, whether something is extruding from the nanodisk, whether you have an incorrect incorporation, a partial incorporation, then the diameter changes, and the complex elutes most likely earlier than a fully integrated protein. But you cannot tell anything about oligomerization from the size exclusion profile in that case.
You are using MSP nanodiscs here, right? I know that there are different MST proteins to do so. How do you decide for the correct one, or is there a default one you resort to?
Yeah, we follow this strategy quite some time. At the beginning, we made certainly a screen of different nanodisk sizes, which are formed, of course, by the different MSPs. And this MSP E3D1 turned out to be, for the GPCRs, the most efficient for integration. Smaller nanodiscs like MSP1, we had a much lower incorporation or solubilization efficiency, maybe with the same membrane composition for the GPCRs. For other membrane proteins like proteorhodopsin, for example, we use different size. So the smaller MSP1 nanodisc is obviously is at least as good as a larger one. And even larger nanodiscs, MSP1, I think N3, turned out to be not very stable in our cell-free system. So the MSP1 E3D1 10 nanometer diameter, obviously, is for that project is the best choice.
This question was, I think, slightly answered already. But anyway, I will ask it again. Which GS proteins was used in this context? Is there a need for nanobody 35 to stabilize the complex?
Yeah, so for the beta 1 and the histamine structure, we use G alpha S. For the free fatty acid receptor, we use G alpha I. And the G protein trimers are not self-free synthesized. We cannot make the G beta gamma in the self-free system because this complex is completely insoluble. So the G proteins, G alpha we could make, but doesn't help us. We need beta gamma as well. So this the heterotrimer, the G protein heterotrimer, we isolate traditionally from insect cells. And as I already mentioned, the nanobody 35 is known to stabilize G alpha, G beta complexes. Again, whether it was absolutely essential, I cannot say because we never tried to make the structure without the nanobody 35. It was helpful for purification and it can easily be synthesized in E. coli. So why not adding it?
Then thanks again for your great talk. And someone was wondering about the orientation of the GPCR when nanotransferred into the cells. Also for human GPCRs, do you think it would be more helpful to use a eukaryotic CFPS to achieve better folding efficiency and function of GPCRs?
For the orientation of the nanotransfer, if you have a symmetric GPCR, so having a similar length of the N and the C-terminal domain, so finally orientation after transfer is about 50-50, 50% right side in, 50% upside down, basically. This is a disadvantage of the nanotransfer. You can modulate the orientation if in that case, at the N-terminal end, you attach a larger hydrophilic domain, or GFP or whatever, because then the alignment of the nanodisc with the cell membrane is favored in the right orientation. So basically with the C-terminus towards the cell. But if you don't have this, you more or less can calculate about a 50-50% orientation. And for the production with eukaryotic cell-free system, I think it's very important, yes, that you have first a decent yield of your GPCR. So our benchmark in the PCF mode is at least, at the beginning to have 1 mg per ml, because during the process you still lose a lot of protein. If you implement this nanodisk strategy, you already have only half of the production, because as soon as a nascent protein chain inserts into a membrane, you have a negative feedback on the speed of the ribosomes. This is also known from in vivo. And also during the purification process, you lose a lot. Yeah, so not to have the requirement to make buckets of cell-free expression. Yeah, I mean we start with 2 mils. Yeah, which is still I would say affordable. Yeah, then you need a good expression system. And whether the folding is better in the eukaryotic system, I'm really doubtful. Yeah. Why should it be better? I mean, we have the perfect quality. I don't know any GPCR structure of a eukaryotic cell-free protein synthesis system.
Yeah, the next question I will just read for now. It will not appear on the screen due to some technical reasons. It is known that the GQ11 class of G proteins is difficult to express and usually requires chaperone RIG8A. Have you tried the cell-free synthesis of the GQ couple receptors, and was it successful in getting a cryo-EM structure of the complex?
No, so only structures we have up to now, I have shown you. We are working on other ones. So again, it's a G protein, so the GQ. We do not synthesize in the cell-free system because the beta gamma, G beta and G gamma, are completely insoluble. For GQ, usually a fusion between GQ and GS is used, but as we have to isolate G beta and gamma anyhow from insect cell cultures, so we already isolate the complete complex. The receptors, so GQ coupled GPCRs, are certainly expressed in our system. So this does not make a difference. Yeah. But the G proteins we obtain from traditional, well-established insect cell culture expression procedures.
The next question is, does it also work with copolymer nanodiscs like SMALPs?
Yeah, so SMALPs are negatively charged and basically traditional SMALPs are toxic to our system because they bind magnesium, and magnesium is a critical component of the cell-free system. However, we can use neutral SMALP, let's say, derivatives. So the so-called sulfodipmalp is also useful to synthesize functional GPCRs. This we have published in, I think, last or 2 years ago, together with a group from Switzerland.
Yeah, for now, we will close this question sectioning. If there are further questions which were not answered, they will be sent per email to Dr. Frank Bernhard. So therefore, they are still answered. And again, I want to thank you for this great talk.